Thanks very much, Claire, and thanks very much for the invitation to speak today. So as Claire's just said, I'm James Shepherd. I'm a clinical PhD fellow here at the Centre for Virus Research, and I'm just going to give an overview of the work that we've been doing here on sequencing SARS-CoV-2 as part of the response to the COVID-19 pandemic. So I work here at the Centre for Virus Research, which is an MRC-funded virology research institute, which is hosted at the University of Glasgow. And we're based on this nice campus just on the outskirts of Glasgow. And I've been lucky enough to benefit from MRC funding, which has enabled me to take 3 years out of my clinical training to study virology. And my interests were, before the pandemic, were in emerging viruses such as SARS-CoV-2. And my research was focused on emerging viruses in Uganda using a technology called next-generation sequencing. And to do that, we set up a clinical study at a site in northern Uganda, where we took blood samples from patients presenting with unidentified acute febrile illness, and used this technology to try and detect viruses that might be causing them to become unwell. And we're interested in what was causing people to become unwell, but we're also interested in animal viruses and things that might be spilling over from the local wildlife. So we also sampled, did ecological sampling, and that included wildlife such as bats and rodents, collected mosquitoes, and also livestock. So I had some experience of emerging viruses and the use of sequencing technologies to detect them prior to COVID-19. So just to make it clear what the viral genome sequences can tell us, so a genome sequence is made of 4 nucleotide bases that code for amino acids, which are the building blocks of proteins. So if you understand the genome or the genomic sequence of an organism, such as a virus or humans or bacteria, we're able to see what amino acids are made, and then we're able to have a good understanding of what proteins that organism makes. So it tells us the instructions for how to make the virus, and it enables us to understand how it works. The flow of information goes from the genome sequence to amino acids and then to protein. And this is a visual representation of SARS-CoV-2. And for example, these knobbly bits on the outside are the spike protein, and these are coded for by amino acids and the DNA genome sequence. So over the last 20 years, generating genomes from humans or bacteria or from viruses has become a lot easier because of the advances in sequencing technology that enable us to generate whole genomes from pretty much any biological material much faster and at a much cheaper cost than it used to be. So if you look back at about 2001, when the Human Genome Project was in full swing, they were using older technologies such as Sanger sequencing. But over the next 20 years, new technologies from companies such as Illumina and Oxford Nanopore have meant that this can happen a lot more quickly, and the cost is a lot less. And that's not just had implications for human genomic research, but it's also spilled over into pathogen genomic research. And these technologies are called next-generation sequencing, just because of the advances that they give over previous methods in speed and cost. As an example, back in 2002, when the first SARS virus emerged, it took about 5 months for us to generate a whole genome sequence for the SARS-1 coronavirus. Whereas on this occasion, when the virus emerged in December 2019, by the start of January, so in under 2 weeks, Chinese researchers had managed to generate the full sequence of SARS-CoV-2, or the virus that causes COVID. So we're really benefiting from this technology. So the initial genome of SARS-CoV-2 was shared online on the 11th of January. And because the genome contains the instructions for creating the virus and for the proteins it produces, researchers were able to right away look at this genome sequence and begin to generate response to the epidemic, such as vaccines. So once they had that genome sequence, that allowed vaccines and other countermeasure development to start right away. And the genome sequence showed that we were dealing with a coronavirus. It was very similar to the initial SARS coronavirus. So this is an evolutionary tree, which we call phylogeny. Once we have the genome, we can compare the genome to genomes from other viruses and see where our SARS coronavirus genome fits into the kind of viral tree of life. And so this phylogeny is showing that SARS-CoV-2 groups very closely with some bat coronaviruses. So that gave us an idea that it probably spilled over from a bat reservoir. And then it's also quite close to the original SARS virus. So that gave us a warning shot that this might be something that could spread from person to person and cause people to become unwell. Now, because I had experience in next generation sequencing from my PhD project prior to COVID, I was redeployed to work on COVID-19 genomics rather than being redeployed back to the hospital like a lot of my clinical research fellow colleagues were. And by March, it was becoming pretty clear that a pandemic was underway and it was going to come to the UK, and it was going to be quite significant. So in response to this COVID-19 Genomics UK Consortium was set up, and that was funded by UKRI, also some funding from Wellcome, the Department of Health and Social Care, and from the Sanger Institute. And the goal was, these were the goals really, to understand the national and international epidemiology of COVID-19, to determine nosocomial or hospital or healthcare associated transmission, to monitor the effects of interventions on the virus genome, such as lockdown or vaccines, and other treatments, such as drug treatments like remdesivir or convalescent plasma. And these goals were all hopefully to be achieved by performing large-scale genome sequencing from COVID-positive patients in the UK. This brought together multiple academic partners and NHS partners from across the UK, and it's actually been very successful. So since we started in March, we've generated over 200,000 genomes, whole genome sequences of COVID from individuals that were affected in the UK, and that gives us a kind of unparalleled understanding as to how the pandemic has unfolded and how the virus is spreading. And lastly, I should say that all that data is provided open access, so researchers anywhere in the world are able to access that data, and they can access the genome sequences that we're producing, and they can use them for analysis. Kind of generating this data, but anyone across the world can use it open access. This is just some artwork from an article that's on the COG website describing the process that we've gone through to get it set up. So if people are interested in COG, I would recommend you go to the website and have a look and read some of the articles there. These are just some of the partners that were there at the start of COG. There's many other partners joined over the last 10 months. But it really does, there's partners from right across the UK. So how do we generate viral sequence data or whole genome sequences? So it all starts with a patient who comes unwell, develops symptoms, and these days will arrange for a COVID-19 test to be taken. So when you go for your test, you'll have a swab, and that will be sent to one of the large kind of mega labs, these lighthouse labs, or an NHS diagnostic lab. And that will be tested using a PCR test, which is there to detect virus genetic material in that sample. And if it's positive, it's because we've detected virus RNA there. So a proportion of these samples containing virus in the viral genome are then sent to a COG-UK site, such as here at the Centre for Virus Research, and they undergo whole genome sequencing. And this is just a map demonstrating the proportion of positive sequences that have been sequenced, positive samples, sorry, that have been sequenced at various locations across the UK. It's a bit patchy just because of the volume of positive tests and the sequencing capacity there, but overall we've sequenced between about 10% of all positive PCR tests in the UK. So It's kind of an unprecedented depth of genomic information we're getting from this pandemic. So once the swab with the virus and the genomic material arrives, the sample is inactivated here and the viral RNA is extracted. The nucleic acids in the virus are then fragmented and chopped up and run on these sequencing machines that generate the data as chunks of viral sequence. And these chunks then need to be assembled into the full 30,000 base pair long viral genome. And we do that by computational or bioinformatic methods here. And then once that processing is finished, it gets submitted to a central computer cluster, which is, I think, hosted in Birmingham, Cardiff, and is also funded by MRC funding. And the computational pipeline there analyzes thousands of these sequences every day in parallel, and then generates analysis that can be fed back to public health agencies and NHS organisations, and can be used for various scientific analysis. And this whole process can be done in less than 24 hours if we need to get an answer very quickly about an outbreak or something like that. And it's important to note that there's multiple people involved with various different skill sets, right from the sample collection stage all the way up to this analysis pipeline. It's a really collaborative effort to get this working. So in addition to the virus genome, we need to get clinical data to put it into context and to really get the most out of the information we're producing. So we want to be able to link the information we get from the genome to the individual patient. So we collect data on things like age, sex, the date we sampled, the swab was taken, things like travel history. In Glasgow, we've gone a step further, and we also have been recording quite detailed information on patient outcomes. So, obviously, some people will have a very relatively mild illness, others will have more severe illness and be required to be admitted to hospital and may even die. So we want to be able to look at determinants of that outcome in the sequence data. So we record that for every patient we get a sequence for. Once we have all these sequences, there's lots of things we can do. So we can use them to design countermeasures such as vaccines and treatments for COVID. We can look at the evolutionary rate of the virus and how quickly it's evolving if we have lots of samples. We can look at that and how it's spreading through populations. We can estimate what the reservoir hosts are. So does it come from bats or pangolins? We can do that kind of analysis. And we can look at the response to the virus genome to therapeutics and vaccines. So do we see a change in the virus that's circulating when something like the vaccine is introduced? And does that have implications for whether or not the virus will continue to work? And we can also use it to investigate transmission clusters and look at determinants of disease severity within the genome. So if someone has a particularly severe presentation, is there something in that person's viral genome that's going to tell us, gives us clues to why they've not done as well? So what we look for are changes in the virus as it replicates and spreads through the population. So every time the virus infects a person and replicates, there's a possibility that it might make a mistake in copying its genome. And if these mistakes are compatible with the virus continuing to be able to replicate and transmit, they become fixed in the genome and they become mutations. So here, if this person is infected with this virus containing this particular genetic sequence which codes for these amino acids, if we see a mutation in that, that will go on to be transmitted to the next person. Some mutations just change the genetic code itself, so the nucleotide sequence, but don't make a change in the protein sequence. Whereas other mutations may change the nucleotide sequence such that it causes a change in the amino acid sequence it's coded for. So some mutations are kind of silent, don't change the virus phenotypically. Others might change the protein sequence and change how that virus behaves. So we're looking for both of these mutations and we can use them in different ways. So understanding the genetic code can also lead us to understand what changes in the protein we're seeing as the virus travels through populations. So because mutations can happen rapidly, so on average, twice a month we see a mutation in the SARS-CoV-2 genome. We can use them to reconstruct transmission networks as the virus travels between different people. And this is a technique called genomic epidemiology, and it's previously been applied to other outbreaks such as Ebola or Lassa fever. This is an example of a phylogeny from the Zika virus outbreak. And we build these phylogenies, which are essentially, it's an evolutionary history of the virus, by aligning viral sequences derived from multiple infected individuals to create this visual representation. So if you imagine that these 5 A, B, C, D, and E are genome sequences from the virus from different individuals, and the colored squares represent mutations. So if we were to take sequences A and B, they both share this blue mutation and this orange mutation. So when building the phylogeny, these two sequences and the virus from these two individuals would be grouped together. And these little tips of the phylogeny represent those sequences. When you look at sequences C, D, and E, they don't have the orange mutations, but they all do share this yellow mutation. So they would be grouped together on the tree. So, and then going on from that, D and E have this green mutation and they would cluster together. So then we can infer that the virus from person E, because it shares these mutations with D, is most likely related to this virus D. Whereas it's less likely to be related to virus A, which doesn't share these green and yellow mutations and has these additional orange mutations. And that's how we build the phylogeny, but we do it on a bit of a larger scale. So we wanted to apply this genomic epidemiology method to investigate how SARS-CoV-2 entered Scotland. We'd managed to sequence quite a high proportion of the cases in the first wave, so we had roughly 50% of all the PCR positive results we had a whole genome sequence for. And you can see in the first wave in Scotland kind of started at the end of, let's go back, started at the end of February, and then by the beginning of April, at the end of March, we were experiencing about 350 PCR positive cases every day. And we had quite detailed information via electronic patient records and specimen request forms on whether or not these individuals have travelled or not. We had information on the dates of sampling. So we performed a combined analysis using the genome sequences that we generated, the dates of infection, and this epidemiological data that we had from individuals such as travel history. And we generated a large phylogeny using all the genome sequences that we could find available to us of SARS-CoV-2 leading up until the end of March. And then we did an analysis of that. So we generated an enormous phylogeny which had in excess of 20,000 sequences, and it's far too big to display properly on a PowerPoint slide or really on any slide. And we did that up until the end of March, and then we added our Scottish sequences on top of that, so our 1,300 Scottish sequences. And we looked at where the Scottish sequences were popping up on the phylogeny, and looked at how where they were in relation to other sequences from the evolutionary tree from around the world. And where they clustered, where they emerged, from the other sequences, we call that an introduction into Scotland. So we're identifying a unique virus with its own mutations that's been introduced into Scotland from the wider pool of circulating viruses throughout the world. And we term these introductions Scottish transmission lineages. So we had some where we had evidence of one individual bringing the virus into Scotland or being infected, but no onward travel. And then we had other examples of lineages where an individual may have brought the virus back and it's resulted in onward travel, onward infection in other individuals in Scotland. And then we had some cases where we had a good travel history of the individuals. So, for example, these viruses were sampled from individuals that had travelled to Italy, and then we're seeing, based on the genomic data, evidence of onward transmission from them within Scotland. And so we used a computational process to go through this enormous tree and pick these out. And what we saw was that there were 234 distinct phylogenetic lineages in Scotland in March. So we think 234 viral introductions into the country. 93 of these resulted in large transmission lineages with multiple sequences, and we could annotate geographical data onto the tree and combine that, and some of these looked quite geographically clustered. So for example, there was an outbreak in Shetland, and Genomics showed that all these cases were related to an introduction to Shetland and one introduction lineage in Scotland. We saw other, so for example, the gold is represents Shetland. We were also able to identify other cases that were linked to this cluster but didn't reside in Shetland. And there were other larger clusters which were linked by Genomics but were more distributed across Scotland. And again, the genomic sequencing allowed us to link these cases together and understand how the transmission is unfolded and show that this was the result of one viral introduction. So 114 of these phylogenetic lineages we were able to determine a known link to travel, so a travel-related case. But for the other ones, we weren't able to identify any known link to travel. So that suggested to us that people were travelling back to Scotland and spreading the virus, but not being identified by the screening measures that were put in place at the time to test symptomatic individuals. The other interesting thing we found was that most of the travel was from Europe, and that there was hardly any introductions of the virus from China. When we looked at the phylogeny, we also found that there wasn't really any genomic evidence of introductions directly from China. So we think that might reflect that there was good travel restrictions related to China around this time, but there wasn't such good messaging of travel or restriction of travel around Europe. So it kind of shows that the outbreak that we have here in Scotland and the wider UK is really a European outbreak, and it isn't directly related to the outbreak in China. So as far as it's come to us via other European countries mostly. We can also use a special type of phylogeny called a time-scaled phylogeny, where the x-axis, instead of representing evolutionary distance, represents time. And we applied this to all of our lineages, and we were able to estimate when we think the virus was first introduced into Scotland. And looking back at them all, we think that the virus first came to Scotland, or there was a common ancestor of all these sequences in Scotland around the halfway through February, which is a couple of weeks before it was initially detected, and suggests it was circulating in the population and evading detection for several weeks before we saw the first case. So once we understand the lineages that we have circulating, we can follow them up over the next few months, and that's what we did using genomic sequencing of positive cases, and assigning each sequence to one of the lineages. And we can also identify new viral lineages when they appear. So thinking back now to August, the city of Aberdeen was placed in lockdown because there was a large outbreak there. And we did some sequencing of those cases, and we were able to show that the virus that was introduced represented a new lineage that we hadn't seen in Scotland yet. So this wasn't a case of spread from the first wave that had popped up again in Aberdeen and caused this outbreak. This was something that had been introduced from abroad. And we could tell that because it was so different to anything else we'd seen in the country before. There was 8 mutations. So that kind of gave us a warning shot as to the role travel might play in driving the second wave. And what we see when we plot the size of the lineages on a plot, so if we look at the time, there's time on the x-axis here, and on the y-axis we have the cumulative sequences, and each line represents a lineage. See in the first wave, the lineages grow very rapidly, but as lockdown occurs, the rate of growth slows, and they kind of peter out in the summer as lockdown takes its full effect, and they actually start to die out. Not many of the lineages actually sustain transmission in Scotland over the summer. But what we see when the second wave begins at the start of August is that these are new lineages that are being introduced from the country, not something that we've ever seen before in Scotland. And these are related to travel. So the genomic data here is showing us two things. It's showing us that the first lockdown was really very effective in reducing transmission. And we almost managed to eliminate the virus in Scotland, and you see these lineages just dying out. But once things open up, introductions of new lineages from abroad start to fuel the epidemic, and that's kind of how we got to the situation we are in now with another lockdown. So given how it's quite topical at the moment, I'm just going to talk a bit about how some of the ways the genomic data has been used in identifying the significance of various mutations that have arisen during the pandemic. So most of the mutations that we have gathered a lot of interest recently are directed at mutations in the spike protein, which is this protrusion you see from the virus whenever you see it photographed or on television or the news something. This spike is the one that we're interested in. And it's an important protein because it mediates entry into our cells. So when the coronavirus infects someone, it gains access by the spike binding onto a receptor on human cells called the ACE2 receptor. And it's also important because the spike is a major target for our immune system, and specifically it's a target for proteins that our bodies produce in response to vaccination or infection called antibodies. And these antibodies that are produced in response to a vaccine or infection bind to this spike protein, block it, and they stop the virus being able to enter our cells because it's covered in antibody. So it's an important protein for these reasons. So back in the summer, there was evidence that a particular mutation in spike called D614G was coming to high frequency in the UK. So mutations are described by their position in the protein. So the spike protein is 1,270 amino acids long. So this nomenclature here suggests that the D amino acid at position 614 in spike has been replaced by a G. So if you hear these names like N501Y or whatever, that means there's been a replacement in amino acid at that particular site. So in March, at this site, about half the isolates we saw had this D amino acid, and the other half had this G, which is D stands for aspartate and G is glycine, so two different amino acids. What we saw as the months went on is that the Ds were completely replaced almost by the G. So people started wondering, is the G giving the virus an advantage in making it spread better, and is that having an effect on clinical outcome or transmission? So because we had we linked all the sequences we've done back to the clinical outcome of patients, we were able to take a good look at this. So we were able to stratify all our patients based on whether or not they had the D or the G at this position 614. And we were able to look back and see what their clinical outcome had been. So had they died, had they required intubation and ventilation or high flow oxygen, had they required hospitalization for oxygen, or did they get on fine and not require any respiratory support? So we did an analysis of this, and it showed that there was no effect of this mutation clinically. So patients weren't becoming more sick because of it. And you can see in this graph that as these are the age groups. As you go up the age groups, the main determinant of doing badly and having a poor outcome is actually age. And that's quite clear in this visualization, and that age is the main thing. But there wasn't any significant difference between the two groups with D or G. So that was quite reassuring. This wasn't making anyone more sick, but it did appear that the virus spread slightly better if it had the G. So this is an example of how we can link clinical outcomes back to a genome data to answer questions. Now, some of the recent mutations that have arisen in the spike protein are of more concern, and there's a particular part of the spike protein called the receptor binding domain that has been a focus of a lot of attention, and this particular domain is important for two reasons. This is a part of the spike protein where the virus binds onto our human cells by binding to this human ACE2 receptor. So it's very important with regards to entry. So we're very interested in any changes in this part that might result in it entering the cells better, more efficiently, or less efficiently. And it's also the most important part of the spike protein for recognition by these proteins I mentioned before, which are induced by infection or vaccination, called antibodies. So antibodies, the most important antibodies our body produces bind to this receptor binding domain. So any changes here might alter entry into cells, and it might also mean that the virus that has a change here might be able to escape recognition by antibodies. So that would obviously be concerning. So just going back to the flow of information, if you remember that we have DNA or RNA in the case of the virus, which codes for amino acids, which then can code for the protein, so like our spike protein with its receptor binding. And if there's a mutation in a certain part of the genetic code, that might lead to an amino acid change, and that might lead to a change in the protein, so change in shape of the protein that might change its function or mean it's not as well recognized by antibodies. This is what we've seen with the variant that emerged in Southeast England just before Christmas and was responsible for the new lockdown. There was a change, an A to a T, a part in the code for the spike protein, which changed an amino acid, which has probably changed the shape of this receptor binding domain and led to it spreading more quickly. So we had a bit of experience with this because we saw a similar change in Scotland back in April, in spike again in the receptor binding domain at position 439. And this emerged and spread in Scotland, caused this lineage here, which reached quite high frequency, but was eliminated because we did a lockdown. But what we've seen is that this mutation has occurred again in several other lineages, which are spread in England, Wales, Ireland, and various other parts of Europe. Because we had identified this by sequencing, we could look at this in the lab. So we went into the laboratory and did antibody binding assays, looking at the two different types. So the N, so the wild type, at 439, and then also the K, so the mutant at 439. What we found was that the mutated form didn't bind antibodies as well for people who'd been infected before. But it didn't lead to any worse infection, and it didn't lead to any increased spread of this mutant. So it just meant that people's antibodies didn't bind as well. So further work's going on at the moment to see if that will also be the case with vaccines. So the concern is because the vaccine contains the code with this variant, if people are infected with this variant, the vaccine might not work as well. But further study in the laboratory and also in the real world, once we start vaccinating people and monitoring what viruses any people who've been vaccinated become infected with are going to be what really tells us as to whether or not this is significant. And something that a lot of people are concerned about are these new mutations in the receptor binding motif. So when we looked at N439K, there was a moderate reduction in antibody binding, and it's not something that we're hugely worried about. But these other lineages have arisen. So there's the lineage that caused Christmas to be cancelled, essentially, which has this N501Y mutation. And then there's other mutations which have occurred in Brazil and South Africa. So that's why we've instituted travel bans against these countries. And it's not because we know that there's definitely going to be an impact on the efficacy of the vaccine, but it's basically being very careful because we don't want these spreading here. And there's evidence that some of them might be able to spread better than the current variant, similar to the one we saw over Christmas. So more work's going into this. There's just been funding from UKRI for a consortium to look at these changes rapidly in the laboratory. It's called the Genotype to Phenotype Consortium. But COG-UK will still be important going forward in identifying where they're emerging, how fast they're spreading, and feeding into laboratory and clinical studies. So in summary, that was just quite a fairly broad overview of what we've been doing. COG-UK has been really successful in generating a really good understanding of the genomic patterns of COVID and identifying mutations that have emerged as the epidemic's gone on. We can understand transmission patterns with genomic epidemiology. It's showing that lockdown is successful in stopping transmissions. It's demonstrated the role of travel-related introductions, which we think are a major driver of the second wave. And COG-UK will continue to look for new variants as they emerge and as vaccines are rolled out. And these are just some acknowledgements, but there's just an enormous amount of people who've collaborated on this, and it's been really, I guess, it's been really inspiring to work in this way, where people have been so enormously collaborative and worked really hard. And a lot of people haven't had much recognition for that, but people have really pulled out old stops in the last 10 months to make this work.